avoiding heat. Alright? Okay, and 3, 2, 1, go. Who captured any? Again, we'll try one last time. 3, 2, 1, go. Alright, so we're gonna start our afternoon. The madam here. And I'm going to welcome Professor Ann for her talk, and hopefully we're all rejuvenated back from lunch. Karibu.
So, asante sana kwa chakula for the lunch.
Thank you for the
perfect.
For the food. Yes.
So I would just say I would like to really thank the organisers for this fantastic opportunity to be here on your inaugural conference. So I'm now hoping that the amazing people who do the AV will bring up my title. But while they're doing that, in order that they keep on time, and I know Prof is doing an amazing job to keep us on time. Right, there it is. So, my very good friend and colleague, Professor Mashidi, gave me the talk to talk about collaborative physiology research within and beyond Africa, which I thought was extremely challenging because, I haven't really thought about this category, but then I thought about it for a little while, and so I found a way that I think I'll tell you about the science that I've learned, and also, hopefully, actually come back to the collaborative physiology research and actually catch some of the ideas that were presented earlier. And really, I want to tag along on the two talks that were just started. And Dr. Abdulatif, who really said, is physiology important? And I want to agree with him and say that it is indeed really important. And also, Professor Essel, Professor Judith Zira, who I share the lab with, and who was actually up here before because she has been one of the members of the Nobel Committee. She's still on the Nobel Assembly, but she's not on the Nobel Committees. And so, I want to take you a challenge that was really posted by Professor Essel, when he said, you can study something boring, like exercise. Now that's what I studied, and I'm trying now to take this challenge and prove to you that it's not boring at all. Which is and a challenge, and it's now just after lunch, it goes practical. So let's see if we can actually get this to work. So the example of an exercise fact, and I'm only going to talk to you, well not only, I will talk to you a little bit about it, but about skeletal muscle. So why skeletal muscle? And this is just now a reminder, sort of a bit of Physiology 101. And that is that now, as we all sit here and we have eaten and it's postprandial, where is our glucose going to be disposed? Here we have an insulin release, and here is a very old study. Now, some of the people in the back of the room, you weren't even born when this study was done. But here it's where glucose goes after a meal. And here at the bottom is the brain, so that's always receiving a lot of glucose. This big yellow part is skeletal muscles, that's what I'm going to talk about. And the reason is not just that it's the biggest glucose disposal organ, but it's also, in fact, here on the right, we can see that if you have type 2 diabetes, that's when the glucose is not growing anymore. So there's a significant impairment in the ability of skeletal muscle to dispose of glucose in people with type 2 diabetes. And just to then remind you, it's also a very large organ. Here we have sort of just one person. Most of us, we have between 40 and 60% of our body is skeletal muscle. So it's a significantly large organ. Now, why am I going to talk to you about exercise? Well, over here, you see what I'm saying. So why exercise? Well, again, we're back to a paper that's published in 1988. And what you see here is a glucose challenge curve. So basically what you do when you want to prove that someone has diabetes, so basically see how well does your body handle glucose. You give a glucose challenge in the fasting state and you follow glucose excursion over 2 hours. And the curves here, the one at the top, is in 7 men with mild type 2 diabetes at the start of the study. And the study was a 7-day exercise intervention. And after 7 days, the investigators did the same again. And here is a glucose challenge curve. after 1 week of exercise. And you can see what the intervention was. It was 6 days of supervised exercise, 1 unsupervised, pretty intense exercise. But despite the fact that we now have semaglutide, SGLT2 inhibitors, all these things, there is really no medical treatment today that has this kind of efficacy. So why don't we use it more? This is a question more for students of psychology, human behavior, I think. But clearly we know this works. It doesn't work for everybody. This we will get back a little bit to the idea of precision medicine. In these ones, it worked in 5 out of 7. But you still got the stats. And in 1988, you could actually publish papers with just 7 people. It's not really possible anymore. And the reason why it only worked and why it didn't work in 2 of them was that they actually already had the beta cell failure, so they couldn't improve their insulin sensitivity. Because that is what happens. In response to an exercise, what happens in your muscle? Well, acutely now, if we had exercised a bit more when Thomas was getting us through all the hand working, what happens in the exercising muscle is that you have an acute uptake of glucose, even in the absence of insulin. You have acute muscle glucose uptake. But you have a lot of other things as well. You have a disruption of homeostasis, and we have the increased energy demand, and we have enhanced insulin sensitivity. And even one single exercise bout will significantly improve your whole body insulin sensitivity, or actually it's only in that muscle that exercise, really, for several hours afterwards. And then you have the chronic effects. That's what I'm not going to talk about. We'll talk about the acute effects, but chronic effects are somewhat the sum of many, many different small acute exercise bouts. So if we want to look ahead, so we already knew this in 1988 that exercise is a highly efficient way of improving your insulin sensitivity, of improving your type 2 diabetes. So what are the challenges? Now, in contrast to Dr. Karolinska, I'm not a purebred physiologist. I just started out as a molecular biologist, and I've spent the past 20 years trying to become a physiologist. But if we just put the question that I've been trying to study for a long time is, we have muscle here on the left, and we have the trained muscle, which has more increased insulin sensitivity, strength, endurance. It's metabolically flexible, means it can use lipids even when you are performing exercise, and has an increased number of mitochondria. And the only input is training. The input is training. So what is training doing? So clearly it has to do something to remodel the exercise metabolically and functionally. Well, no, it remodels functionally, because if we want to run a marathon, we all train, because you have the functional remodeling, but you have a metabolic remodeling as well. How does it happen? Well, it could be transcription factors, could be different metabolites, histone modifications, DNA manipulation, non-coding RNA, and that's basically what I want to tell you about. How are we trying to understand that? But first, I want to give a little vignette from one of my colleagues, Nico Pilon, and just show you the power of many small studies and how they can work together. And what Nico did, he's also works with exercise physiology. He mined the data, the public data that's available on human exercise intervention trials, and basically downloaded all the data that's there. It's free, it's out for all of us to use. But what Nico has done, he's made a database with all these various studies. And so it's also available to anyone who wants to use it. So write down metamex.eu, because you can use it for your own favorite gene. But what Nico did was, he basically looked at all these acute aerobic studies, training aerobics, so when you do many exercise bouts, acute resistance, or training resistance, so more like hypertrophy, or inactivity studies. And he put them all together, and he basically cleaned the data up so that you can compare it. So actually, if you go online on the metamex.eu and you put in your favorite gene, you can then select, I want to only have the acute exercise studies, or only the acute resistance studies, and it will tell you if your gene is modulated. And just to tell you a little bit, if you just do the global whole picture analysis, here's the principal component analysis, and what you can see here, the orange is the acute resistance, and the yellow is the acute aerobic. And you can see that they segregate away from the training effects, which are more over here. So it's more the duration of the exercise than what kind of exercise you did. But anyway, it's just a little plug to say. Now here's just to remind you of the fact that when you do exercise and you move your muscle, clearly a lot of different systems are activated. And here's just from a recent review written by my colleague Janine Zira of all the things that we already know are activated. But the question is, how is it cognitively regulated? So now I want to take you through an ongoing study that's going on in our laboratory at the moment. Some of it's a little bit published, but really it's all based on one sort of study paradigm. And I've just divided it into three. It's muscle exercise changes in mRNAs, in skeletal muscle adipose tissue crosstalk, and a little bit about metabolomics at the end. So this is the setup that we use for all these three studies or vignettes. And we've recruited people who either have normal glucose tolerance or have type 2 diabetes. And they come into the lab in the fasting state. And we take a biopsy. And then they get on a bike and they exercise for 30 minutes. And we've actually screened them a little bit earlier so that we know exactly what's 85% of their maximum heart rate. So they do that for half an hour. They get off the bike and we take biopsies again. And the samples we take are muscle biopsies, fat biopsies, but we do that in the same incision. So on the way into the muscle, we actually take the subcutaneous fat, and then we also take blood. And here is if you have type 2 diabetes, and here you have type 2 diabetes 3 hours later. Now it's a bit difficult to appreciate from here, but what was really striking is that the number of genes that changed in somebody, and it's only men here and this study, the number of genes that changed in men in response to exercise was about 500. It's over there somewhere, 537. The number of genes that changed in someone with type 2 diabetes was 4,500. So a whole different order of magnitude. Which, and this was at the 3-hour time point that we saw this. But it's really surprising, thinking, at some point, what's the exercise doing in a person with type 2 diabetes that's so very different? And this was published last year, so you can, if you're really interested, you can read the paper, all the fancy analysis. But basically, what we see, or what we think we see, is that in the person with type 2 diabetes, it's not evident at the resting stage in muscle, but after the exercise, there's a huge call for inflammatory cells. They seem to be flocking into the muscle and then changing the whole transcriptomic landscape. And we don't know whether this is a good thing or a bad thing. Or quite what the implication is, but clearly it's quite different. And as a matter of fact, we then, Nico, looked using immunocytochemistry at CD11 plus cells here. And what you can hopefully see, it's a bit difficult on this screen, is that in the pre-stage you see very little difference between the diabetics and controls. Post, you don't see very much either, but that 3-hour time point later, we have this infiltration of immune cells. So, here, this is what happens when you steal pictures of the internet. It's been, there should be a picture of Frederick Vasily there. But I was going to say that this increase in its inflammatory tone, we'd already been suspecting for a long time. So when Frederick Vasily was in the lab in Stockholm with us, he did a study, but this is again now just in the resting state, where he looked at the phosphotranscriptome. So he could see that there was an increase in phosphostat-3 and phospho-react-2 in people with type 2 diabetes. And those of you who are not into JAK-STAT signaling, you just have to take my word for it, that's kind of an indication that there is some increased inflammatory tone in the basal state. It couldn't be seen in the mRNA, we couldn't see it there, but here we're looking at the phosphoprotein. So, and I guess that only showed you the men. So at the moment we're trying to study exactly the same thing in women, but we haven't quite got that far yet. But during the same time, another colleague in the lab, Ms. Delay, was interested in using the adipose tissue. So she took the adipose tissue from exactly the same people. She studied both men and women. And so the interesting thing here was the adipose tissue, it's next to the exercising muscle, but it didn't do any work. So it's sort of an innocent bystander, but we wanted to see, was something happening there too? And to sort of cut a long story short, what we saw was a very, very similar difference again. So now I'm just showing you the pre versus post. So here's when in the adipose tissue, the pre versus post, when immediately no one got the bite, there's about 500, 600 genes that change in adipose tissue. But when you look at that 3-hour time point after they got the bite, here's the type 2 diabetes circle again, and it's much bigger than in the people with normal glucose tolerance. The adipose tissue is also responding very differently if you happen to have type 2 diabetes and get on the bike. So, now I go back to the muscle. There. Get this to work for me. Okay, it's just a reminder that's what it looked like in the muscle. So what Lucille did is she was thinking that perhaps what we should really look at is secretory factors and look at what's the tissue prostore. So here we have again the same data pretty much of the difference, but now she's only looking at the genes that encode for a secreted protein. So again, this is all kind of bioinformatics. She's taken all the genes and kind of looked at them and said, what's the signature? And do any of these genes in the adipose tissue look like they would be secreted? And the little pizza pies up here shows that it's about the same in a normal glucose tolerant type 2 diabetics. So here is not, that doesn't explain the big difference here. But she was interested in seeing what's adipose tissue releasing in response to exercise. And there are a number of proteins that have been reported to do that. So we saw sort of the usual suspects, which is always nice. It means that you see what other people see. But what Lucille also found was a number of genes that hadn't really been studied that much before. And in particular, I want to tell you a little bit about this one, which is called oncostatin M. And as you can see, it goes up very, very markedly in the adipose tissue at 30-minute time. And it comes back down again pretty much. So oncostatin M was the thing that we really were interested in studying. And why did we just pick oncostatin M? It wasn't completely random. What we already knew was that if you treat obese mice with oncostatin M, you actually have improvements in adipose tissue inflammation and insulin resistance and a lot of good things. Basically, that had been shown by other people. However, there's a bit of a difference in mice. The oncostatin M is increased specifically in skeletal muscle fibers. Now, because we have the data from the muscle, we knew that in the humans, oncostatin M does not increase in muscle from humans, but it does seem to increase in adipose tissue from humans. And indeed, we then went on to say, okay, the important thing is, so far we'd only looked at the mRNA in an adipose tissue. So what really happens, of course, if we then look to see the business end, we went to the blood samples to see is there really an increase in the circulation. And indeed there was. And interestingly, the increase in the circulation is higher in the normal glucose tolerant people than in type 2 diabetics. And we also saw that there was a sex difference in the response here. So what could it be doing? Well, of course, it's quite difficult to do the mechanistic studies in humans, but we tried to kind of figure out if we took cultured adipose tissue in vitro and we exposed them to a number of different substances, but one of them was oncostatin M, and we could see that we had a quite dramatic increase in phosphorylation of ERK. Now, phosphorylation of ERK can do a lot of things. One of the things it can do is increase lipolysis. And in an experiment I'm not showing you is that this actually does increase lipolysis. Oncostatin M increases lipolysis in cultured human adipose cells. In vivo, the only thing we could do is really go back to the tissue and see, okay, if people exercise, does ERK phosphorylation increase in adipose tissue? And indeed it does. But this, of course, is a little bit hand-wavy. I'm just saying it's correlative. But at least it goes in the right direction. We then went to try and think a bit more carefully. Like I said, we knew that oncostatin M doesn't come from muscle in humans. The other thing that we could see when we now went to the online databases to download all the different kind of single-cell sequences is that oncostatin M is primarily expressed in immune cells. It is not expressed in adipose tissue. So again, we think it's the immune cells that might be coming in to do this. However, the receptors are specifically enriched in the adipocyte fractions. So what we think is happening, and also then we went to see, is this really true? So I'm showing you here some RNA scope
Well, basically, again, in adipose tissue slices, we specifically tried to go in with RNA to see whether we have CD68 as markers of immune cells, and see whether it actually co-localizes with the oncostatin M. And we think that monocytes and macrophages are the likely source of oncostatin M in human adipose tissue. And here's just to prove to you that in the muscle, we really see no oncostatin M at all being expressed. And the receptor goes up a little bit. So, oops, let's skip that one. So what we think is happening is that when you run, you have a signal from your muscle that somehow recruits immune cells, not only to the muscle itself, but also into the adipose tissue, and that this is different in people with type 2 diabetes, and that alters metabolism. So how can you test this? Well, you can't test it in a human, so we tried to do sort of the next best thing. So we took some cultured immune cells, and we treated them with things that we thought would mimic exercise, like clenbuterol for scoring. And what we could see then is that when we do this to the immune cells in the test tube, we can have an increase in oncostatin M. The other thing that we did is we took cultured human muscle cells and we contracted them in vitro, so kind of see whether they produced something. And so we took this media from the contraction, so we contracted them with electricity, and we could then say that we get a peak of the oncostatin M. So we think that there is the possibility of crosstalk. So what we think is happening then is that the exercise induces inflammation in adipose tissue and muscle, and somehow speaks to this factor, increases something in the immune cells, and the immune cells increase oncostatin M, and the oncostatin M change the adipose tissue metabolism. So that's our attempt at looking at all of the crosstalk. Now, before you completely fall asleep, I'll tell you a little bit about exercise-induced metabolomics. And so metabolomics was measured, we measured it both in the plasma, but I'll tell you here what's happening inside the muscle. So now we're back in muscle, in the muscle tissue. And what we then basically looked at untargeted metabolomics, so all the metabolites that change. And there's lots of them. So I'll just walk you through to kind of try and see some patterns. So here it's the female normal glucose tolerant in response to an exercise bout. Especially what you see is branched chain amino acids, you see lysophospholipids and sphingomyelin. So now we're in normal physiology. These are normal glucose tolerant people, and they are women. And that's what happens when you cycle on a bike. If you're a man, it's pretty much the same. Doesn't seem to be a big sex difference. Again, the branched chain amino acids are the biggest thing that happens, and you have a few changes in fatty acids that you didn't really see in the women. Now, type 2 diabetic women. It's a little bit different. We do see the branched chain amino acids. That seems to be like the number one thing that happens to metabolomics when you exercise. But we also see some changes in fatty acids and long chain PUFAs and sphingomyelinases. So that's in type 2 diabetes. And in the men with type 2 diabetes, branched chain amino acids again. But now you have changes in lysophospholipids, diacylglycerol, and then these long chain fatty acids. So I'll just try and summarize it a little bit. So what happens all the time is branched chain amino acids. I didn't show you the TCA cycle, but most acyl conjugates. These things seem to change in muscle when you exercise. Disease-related difference are diacylglycerol and a few glycolysis-related metabolites. And sex-related differences are the ones that are more related to lipids, like long-chain PUFAs, lysophospholipids, and sphingomyelins. But I want to show you this look at it as well. So now here's one of my colleagues plotted this, sort of the change as a fraction of time, because we have the before getting on the bike, getting off the bike, and afterwards. And so you see the amino acids have this kind of peak. It's just grouped on a number of different things. So here again, it's the women who have like a peak in branched chain amino acids, and then it kind of stabilizes. Let's add the men. The normal glucose tolerant men are very similar to women. You see this change in energy, sensing, metabolites, but there's a difference in lipids between men and women. Now we have the type 2 diabetics. So again, the women, we still have this amino acid change, but we have a much more a sluggish response on the lipids. And now we have the type 2 diabetic men. And again, we see that the responses are sort of similar, but they are delayed. And we're still sort of trying to put our heads around this and get this all together. But I think, if you remember, the glucose challenge that I showed you in the beginning is often delayed in a person with type 2 diabetes. And what we seem to see is that it's not just glucose. The whole metabolism seems to be more sluggish. And now we're not going in with a glucose challenge, we're going in with an exercise challenge. And for some reason, the whole response just seems to be different. So now I want to kind of highlight a few things that, for those of you who kind of born with me here now. So this is an ongoing project that we're working on at the moment. We sort of call it multiple omics in metabolic disease, and it's just one single exercise bout. And we've just now started analysing things like DNA methylation. Now, the beauty of the study isn't so much that it's complicated. Of course, we've done every single omics we could think of, which is clearly expensive, but really the importance of the study comes from a very well phenotyped intervention. And this sort of intervention needs to be done not just in one population, it needs to be done in several populations. And perhaps if one can identify what are the key things we need to look at, then you don't have to do all the omics, but you could do the smart intervention in a different population to say, well, is this true? This conclusion that we want to say, which happens to be true for men and women who are about 57 years old and live in Stockholm, how does that actually pertain to the rest of the world? So, now for those of you who were just wondering what I was talking about, the take-home message is that an exercise bout in a person with type 2 diabetes results in a very different response than exactly the same bout in a person with normal glucose tolerance. So that's the one thing you need to write down. And the other thing is that many of these things are not different in the basal state. We know, like I said, we know for 40 years that muscle responds differently to insulin. But when you compare muscle biopsy from people with type 2 diabetes or normal glucose tolerance in the basal state, when they're just resting or fasting, there are very few differences that you could see, especially in things like the transcriptome, which is what we've been studying for a long time. But you stress the system and you can unmask the differences. So, looking ahead. So now we come back to, is physiology important? So what I've tried to walk through is that we can now study on a global omic level things like DNA, we can study DNA methylation, histone methylation, with of course RNA or mRNA transcriptomics, which is something that 20 years ago was very, very expensive. Now this is pretty much routine. It's quite inexpensive to study this. Same with DNA methylation, that is quite inexpensive. However, studying protein and protein modifications is very expensive and very complicated. You need high sophisticated machinery, same thing with metabolomics. And then in the end, you need to have clever bioinformatics. But, so this is where I'd like to kind of come in. Even with the most sophisticated and most fantastic proteomic studies or protein modifications, or the most sophisticated single-cell RNA sequencing, in the end you end up with a list of correlations. And that list of correlations doesn't mean anything until you've translated it into physiology. Because otherwise it's just correlation. So the translation to physiology will require physiologists. And while I think that AI might help us with the bioinformatics, it will still only give us a more clever list. And if we cannot, as physiologists, test the hypothesis, then it will be meaningless. The other thing is, of course, we need to look at the different tissues. And finally, we like to think about this thing of precision medicine. So precision medicine is clearly something that is important, because we know that it's not one size fits all for a pill. We know it's not one size fits all even for exercise. I alluded to that at the start. And we know that for some people, maybe it's a cycle bout that's really efficacious, whereas for other people, it might be that strength training is more efficacious. But we cannot do precision medicine if we don't have multiple different populations. And at the moment, we are, as a sort of, collective, most of the data is from Northern European men. We're not even studying women well enough, and I think Professor Raveley highlighted that too. But we are only studying one small subgroup. And so I think it's really important to see, is an exercise bout in a man in Stockholm the same as an exercise bout in a man in Japan or an exercise bout in a man in Nigeria? That we don't even know yet. And so what I'd like to say at the moment, the low-hanging fruit isn't to do all this exactly the same. The low-hanging fruit is to figure out what is the key question, what is the key readout that you have found? And can you replicate that, or is that different? The other thing that, of course, needs to be done, and one thing that we've been studying is that an exercise bout done in the morning for somebody with type 2 diabetes is less efficacious at reducing blood glucose than one done in the afternoon. But that's only for one type of exercise. We use high-intensity interval training. Maybe if we had used another exercise intervention, we would have seen something different. And then the only readout there was glucose control over the next 48 hours. So that's not a particularly expensive measurement and can be done everywhere. The difficult thing is in careful physiological phenotyping, in the selecting of patient and the matching of patient, in making sure that the exercise intervention is exactly what you want them to do. And that, I think, is something that really can be done even on smaller budgets. It just takes very passionate physiologists to do it. So, even though I think looking ahead, we are moving towards larger omic analysis, but the large omic analysis doesn't have to be done all in one lab. It can be a collective, be it in rodents or in men. And to gain precision, for precision medicine, we need very much to increase the variation of the samples we study. We can't just be studying medical students in Boston. And the future will absolutely require collaboration, because not one person can do all this, not one lab can do all this. And again, I just want to thank all the lab that I share with Julene. And I was looking at the picture this morning, and we actually have people represented from every single continent, although at the moment the African subcontinent is represented by somebody from Egypt, so he looks a bit pale. But, so with that, asante sana. And promise will tell me when I should sit down.
There we go.
Yes.
Thank you. That was quite a lot. Let's do our thunder clap for Professor Ann. Okay, wait. Thunder. And clap. Thank you very much. Okay, so I think we'll welcome our last plenary for today before we get to the questions. Professor Kadiri from Benin. Karibu sana.
This might be working better.
Thank you very much. Okay. Thank you. Maybe I will thank the organizing committee at the end of my presentation. So, thank you. Ladies and gentlemen, that's a part of the result of our lab on the topic beneficial effects of medicinal plants in diabetes, immunological aspects. So, the outline of my presentation is composed of these points. First, I will shed a light on the burden of NCD, non-communicable disease, the children have in the South. And I will focus on the place of diabetes in NCD, the classification, meaning, and physiopathology of diabetes. How does the immune system interfere with the severity of diabetes? And after, I will finish on how do the anti-diabetic plants can regulate the immune system, and I will finish with a take-home calculation. So, the first question on the burden of NCD in the global health. So, who do people die from? The people, if you look at this map, you find that over 74% of the global death are due to non-communicable diseases, and 14% to communicable disease, and 8% to other injuries. And in detail, you find that the heart disease occupied 33%, cancers, chronic respiratory disease, digestive diabetes, neurological disease, and other non-communicable diseases. But in Africa, what we notice is that the big three pandemics in Africa, HIV, malaria, and tuberculosis, just lead to only 5% of death. But the majority of the financing is allocated to the big three pandemics in Africa. And additionally, the study or the research not focused on fundamental research, it's only on operational research. You go and count how many people suffer from HIV or malaria or tuberculosis, but not the fundamental to get the mechanistic research. No. And they throw so many money in this disease, but then led completely non-communicable diseases. And I want to share with you this correlation. You know, the viral disease, the communicable disease, and the non-communicable disease is very high in sub-Saharan countries in Africa and South Asia, maybe communicable and non-communicable disease. And if you merge the both, non-communicable and communicable disease is also very high in sub-Saharan African countries and South Asia. And what we notice is that the high is the burden disease. Yes. Increase the voice. Okay. Thank you. We notice that the high, the income of the country is very low, and the burden disease is very high in this country. So that is a very wonderful correlation. And that is why in this country, sub-Saharan African countries and South Asia, a larger part of the population, over 80% of people rely on herbal profession for their primary care. But so far, the scientific investigation to support the use of these plants are very, very poor in these countries. Now, what's the place of diabetes in NCD? You know, diabetes belongs to the top 10 causes of death worldwide. In the year of 2019, 2.2 million adults died from diabetes worldwide. And in Africa, especially in North Africa, the prevalence of diabetes is increased every year compared to sub-Saharan Africa. Especially in my country, Benin, you know, the prevalence of diabetes in the year 28 is 2.6, and it increased fourfold into 12% just in the year 2016. So that is very, very worrying prevalence. And what is diabetes? Every people, you know what is diabetes. What happened? Although the classification criteria which allow to classify as two major types of diabetes, type 1 diabetes and type 2 diabetes, there are some intermediate forms of diabetes which cannot be classified, like tropical diabetes, also named malnutrition-related diabetes, and gestational diabetes. And what happens? Physiologically, in the morning, after fasting glycemia, after fasting overnight, you know that your glycemia is around 0.7, between 0.7 and 1.10 gram per liter. And in the morning, when you take your breakfast, your glycemia, the blood glucose, increase rapidly and decrease after 2 hours to normal level. And when you talk in the lunch, you take the lunch, as we take just now, the glycemia increase and decrease after. And the similar result is obtained after the afternoon tea and the dinner. Glycemia increase and rapidly decrease after 2 hours, normally to the normal level in healthy person. But in diabetic person, what happens is that after glucose ingestion or food intake, the glycemia rapidly increase in type 1 diabetic patient as well as in type 2 diabetes to almost 3 gram per liter or 4 gram per liter. And this glycemia did not decrease after several hours. That is named the chronic hyperglycemia. And physiologically, what happens? You know, in healthy person, after food intake, the pancreas produce insulin. And this insulin binds to its receptor on the somatic cells in the muscular, adipose tissue, liver, and brain, and allow glucose to enter into the cells. And so the blood glycemia diminished. But in type 1 diabetes, the insulin is not produced by the pancreas because the pancreas is autoimmunity destroyed by autoreactive cells. And no insulin, so no insulin to bind its receptor on the somatic cell, and the glucose remain in the blood. And in type 2 diabetes, the insulin is produced, but this insulin cannot bind to its receptor, and the result is that glucose remains in the blood, and that is what we name hyperglycemia. And this chronic hyperglycemia have led to several disorders in several vital organs, like deterioration of blood vessels, cardiovascular disease, deterioration of nerves, retinal disease, as we name retinopathy, and the kidney, which lead to renal failure. And in gestational diabetes, you know, pregnant women who develops diabetes during pregnancy, maybe during the first or the second or third trimester of the pregnancy, or women newly with type 1 or type 2 diabetes, these pregnant women are at high risk, over 62% to have macrosomic babies. And these babies are the large babies whose birth weight is over 4 kilograms at birth, and this is related to several complications like fetal death, prematurity, and principally these babies are at high risk to develop obesity and type 2 diabetes during adulthood. And they name the phenomenon, physiological phenomenon that is named metabolic memory, is the result of the hyperglycemia that the fetus is submitted during the uterine life. During uterine life, the fetus is submitted to hyperglycemia, and this leads to fetal hyperinsulinemia, and that we name fetal programming. And this fetal programming is conducive to metabolic memory, which induced obesity and type 2 diabetes in offspring during adulthood. And what in maternal diabetes, the hyperglycemia reacts on fetal hypothalamus, which produce a peptide that we name neuropeptide Y, and neuropeptide Y increase the trend of high food intake in the fetus. That is, introduce macrosomia, lead to macrosomia and obesity in offspring during adulthood. Now, how does the immune system interfere with the severity of diabetes? So, in type 1 diabetes is a result of interaction of genetic predispositions and environmental factors like viral infection or bacterial, and this leads to inflammation into the pancreas, which is infiltrated by autoreactive T cells, and these autoreactive T cells destroy beta cell, and beta cell, which is supposed normal to produce insulin. So no insulin, and this leads to hyperglycemia in type 1 diabetes. And what we know is that the all condition that will increase the inflammation in this condition will potentiate the severity of diabetes. And we know that CD4 T cell, and the naive CD4 T cell, CD4 T cells is the category of T cell that's the major conductor of the immune system in humans as well as animals. And these cells can be differentiated upon the presentation of antigen or the environmental of insulin cytokine environmental. These cells can be differentiated into type Th1 T cells or Th2 phenotype. And the Th1 T cell produce mainly IL-2 and IL-10 or interferon gamma, L12 and interferon gamma cytokines, and these cytokines promote cell-mediated immunity, and they are pro-inflammatory, and this is deleterious for type 1 diabetes. In type 2 diabetes, there is insulin resistance, which leads to chronic inflammation in the several organs like adipose tissue, pancreas, liver, and kidney. And this inflammation leads to the infiltration by of all of these organs by autoreactive cells like macrophages or T cells, which produce in situ pro-inflammatory cytokines like IL-6, L1, TNF-beta, TNF-alpha, and these disrupt the functionality of all these organs. So, in gestational diabetes, what happens? It is in normal pregnancy, we observe that the TH phenotype Th2 phenotype increase, whereas Th1 decrease, as well as in diabetic pregnancy. This is due to hormone production. But in macrosomia, the result is not similar because Th2 decrease and Th1 phenotype increase, and as well as in adult obese offspring, the Th2 decrease and Th1 is increased. That is characterized by inflammation in macrosomic babies, and this inflammation disrupts all the organs that we know in gestational diabetes, like placenta, adipose tissue, and leukocyte, which produce adipokines and cytokines, which are which have a pro-inflammatory activity, and this is deleterious for GDF and macrosomia. And all action that can shift the differentiation of T cells into Th2 can be beneficial for the patient with type 1 or type 2 diabetes. And we did a study in our lab, and we found that in type 1 diabetes or type 2 diabetes with insulin, they have Th2 phenotype which increase, so that it will be beneficial. And now, how could the anti-diabetic plant regulate immune system in type 1 or type 2 and in JDM? There are some plants that we use in Africa, that this plant, Momodica charensia or Citina terabicidica or Becranima nitiga and Nuclear latifolia, I selected these 4 plants because we worked on these plants. These plants are empirically used in Africa and so many countries in Africa and South Asia, and they have anti-diabetic properties. So I show the picture of this plant. Maybe you can recognize the plant in your country. This is Oxygenanthera, Nuclea latifolia, Nuclea and Momodica charensia. We focus our attention first on Momodica charensia as this plant benefits of good attention from the investigator. Several studies have reported that Momodica charensia exhibit anti-diabetic and anti-hyperglycemic activity. And there is a study from Ahmed Ehol in India who showed that Momodica charensia extract has induced regeneration of pancreatic beta cell in diabetic animals. That is very interesting. So that is why we focus our attention on this plant. But we don't know the mechanism action of these plants, maybe immunological mechanism in the physiopathology of diabetes. And so that we conduct our study first to evaluate the effect of the fruit juice and leafy stem on the activation or and differentiation of human T cells. That is the first study in vitro. And after, in vivo, we investigate the anti-diabetic activity of Momodica charensia on immunological parameters during experimental type 1 diabetes. And we prepare two kinds of juice, first from the leafy stem, the leaves, as the apple juice, and from the fruits. And we use total juice, or we filter the juice before use. So what we observe, we first perform a T cell proliferation upon the juice, and we add maybe total juice or filtered juice on the cell proliferation. What we observe is that the fruit juice or leafy juice induce cell proliferation without any stimulation by antigen, by mitogen. And when we activate T cells with mitogen, the results are sent because juice induces the proliferation of T cells. But when we filter the juice before use, we observe that the juice inhibits cell proliferation. That is wonderful result that we observe. Maybe from the juice from fruit or from leafy stem, we observe that the juice inhibits cell proliferation. So we are interested by the level, the production of cytokines in vitro, and what we observe that the total fruit juice, maybe from fruit or leafy stem, they induce the production of interleukin-2 without mitogen. But when we activate the cell with mitogen, like PHR, this result are same because it induces the increased production of IL-2. But the filtered juice from fruit or from leafy did not induce the increased production of IL-2. Same result was observed for interferon gamma, increased production of interferon gamma, but the fruit juice induced the decreased production of interferon gamma, especially from fruit juice. We evaluate also, we measure the concentration in the square atom of IL-4, and we remark that the fruit juice did not change the secretion of IL-4, but the leafy stem juice did not change also the secretion of IL-4. IL-10 also, the fruit juice increased IL-10 production, and leafy stem decreased the production of IL-10. This observation appears to be normal because the IL-10 possess regulatory and anti-inflammatory properties. So we conclude that in vitro, Momodica charensia fruit juice, when the juice is filtered, this induced the differentiation of THCL into TH2 phenotype. That is protective, made protective in the treatment of type 1 diabetes. And we use to screen the fruit juice, and we found that when the juice are very, very rich of tannin and flavonoid and coumarin and steroid also, but the filtered juice, the filtration remove from the juice the saponoside composite. So the filtration remove from the juice the saponoside component. So the partial conclusion is that Momotica charensia elicits the following activity. Total fruit juice reveal an immunoproliferative capacity and Th2 phenotype of activated lymphocyte. On the other hand, the saponoside-free filtered fruit juice show an immunosuppressive activity that still exhibits the capacity for induction of Th2 cytokines. This may be helped to inhibit the deleterious effects of autoreactive cells on pancreatic beta cell and protect against type 1 diabetes. And we perform also in vivo study. In vivo, we treat diabetic animal, so we induce diabetes with streptozotocin injection by 5 low doses, and we treat animal after the diabetic injection with fruit juice. And what we found is that on glycemia, we found that the fruit juice decreased the glycemia, the fasting glycemia of diabetic animals. And we perform also, we measure the production of cytokines in vivo, and we found that in diabetic untreated diabetic animals, there is increase of IL-2 and interferon gamma. This means Th1 cytokines, but the juice in animal treated with fruit juice and filtered fruit juice, we remark that the differentiation is shifted toward Th2 phenotype. So that confirms the result that we observe in vitro. So we also perform study on the 3 plant Nuclear latifolia, Ocytenantera, and Bicralima nitida. So we examine the effect of the juice from this plant in diabetic pregnancy. I show here the part of the plant that we use to prepare the juice. First, we prepare the extract with 5 solvents: ethanol, ethyl ester, ethyl acetal methanol, and aqueous. And with that we obtain 20 extracts. So as we cannot investigate 20 extracts, we use to perform first the antioxidant activity of each juice, and this allows to select only 5 juice among the 20 extracts that we prepare. And we perform the effect of this extract, the 5 extracts, on T-cell proliferation. What we observe is that the 5 juice induce or inhibit the cell proliferation, so that they have immunosuppressive action. And after we use this extract, the 5 extract, to treat diabetic pregnant animal. And we found that we measure the glycemia at day 12, day 16, and day 20. And you know, day 16 is 1 week after the diabetic induction. So we found that in diabetic animal that the glycemia decreased significantly after the animal were treated by a plant extract. And the same result was observed on day 21. So what's the conclusion from this study? Momotica charensia exerts anti-hyperglycemic effect in type 1 diabetes and modulates the immune system by shifting T-cell differentiation toward anti-inflammatory TH2 profile in vivo and in vitro. Medication could be used in the treatment of both types of diabetes. And also, we found that the treatment, the specific extract that we prepare from the treatment, oxytenantera, picralima, and nuclear, show anti-hyperglycemic effects in diabetic pregnancy. So I want to thank the personnel of my staff, Professor Cairo, and the personnel of IRD Institute of Research for Development, likely Professor Ashil Masubuji. And I want to express all my thanks, especially express thanks to my boss, Professor Nayuka. Here is my boss. I did my PhD in his lab, and I want to thank him very, very much. And I took this picture in the year 2007, so it means almost 15 years now. So I was very young at this point. Thank you very much. Thank you very much.
The role of
Sorry, I cannot understand the question. Can you move?
M2 and M1.
Yes, yes.
Good afternoon. So my question goes like this. I have said that there are two populations of macrophages in adipocytes. So one is M1 and another is M2. So M1 performs a pro-inflammatory role, while M2 has contribute to inflammation. So did you check on all this expression or expression of all
Some type of macrophage.
or the role they play?
Okay, that question is there. I will take one more and then we can get the speakers to answer. One more question? Oh, there we go.
Good afternoon. My name is Manasseh from the University of Nairobi. Okay. My name is Manasseh from the University of Nairobi. I also have a question for you, Professor. Yeah, so you say the total fruit juice has an immunoproliferative effect on the Th2 phenotype in one of your conclusions, but in the data that you showed us, the cytokines for Th2 are suppressed, so I didn't understand how that comes about.
Okay, I think Professor Kadiri is in a hot seat. All the questions are his.
Thank you very much for the question. That is an interesting question about M1, macrophages 1 and macrophages 2. Really, we did not in this study discriminate between the subtype of macrophages, but as we noticed that in human beings as well as in animal studies, we noticed that ILTs and TNF alpha increase in diabetic subjects, so that we can conclude that it's mainly related to M1. But in this study, we did not discriminate between the both types of macrophages. And the second question related to the production of cytokines, Th2 cytokines. Yes, I say that the total fruit juice or leafy stem juice induce lymphoproliferative action. And the cytokine, you remark that Th2 cytokine decrease or increase? Decrease.
You say the cytokines decrease, yes, but in the conclusion you said total fruit juice.
No. It is Th2 cytokines decrease and Th1 cytokines increase. And this is concomitant with the proliferation of the cell induced by the extract. That's what I'm saying. Yes. Yes. It is very, very coherent.
Okay.
Good afternoon. Good afternoon, everyone. My name is Henry Nyongesa. I'm from Masinde Muliro University of Science and Technology, located in Western Kenya. My first question goes to Professor Kadiri. That was quite an interesting presentation where I did my MSc, my DSc. One of my colleagues was doing actually a study on Momodica Karantia and its effect on diabetes. My question goes on one of your conclusions where you are suggesting that Momodica Karantia could potentially be used for treatment. So I'm interested in knowing, and I think this is something that was highlighted before, the gap between translating basic research to clinical applications. I don't know whether you have any plans to translate in terms of getting the dosage for this Momodica Karantia. How much are people supposed to be consuming? Is it on a daily basis or is it on a weekly basis? Yeah, so, and then my second question goes to Professor Khan. It was quite an interesting presentation with a suggestion about some anti-obesity spray, something like that, yes. So I was interested in, like in an African context where we have, especially in a rural setting where there is consumption of mostly unrefined foods, like raw natural foods, whether they have any potentially an effect on modifying the signaling molecules that you mentioned, like the CD36 and GPR120. Do you think that just consumption of, say, natural foods can maybe downregulate GPR120, and that can actually be like a natural way of just evading from the obesity pandemic? Thank you.
Okay, I think we can have the responses before I take two more.
I can give your response first. Yeah. Okay. Thank you very much. Because yes, I know. You know that Momodica Karantia is the plant almost used by our population. You eat, and everybody eats. They use this plant, and they use this plant for its anti-diabetic properties. But we perform our study in animals, really. Now, we did not transfer to the clinical setting, but the level of this study is that you have to, the fruited juice, you have first the juice from the fruit will appear to be more effective than the juice from leaf. That is the first conclusion. The second is that you have to filter your fruit, your juice, before use. Because if you filter it before use, you have the effect, the beneficial effect. But however, if you do not filter it before use, you can have opposite effect. So that is a suggestion that I can do for you. But we did not, at this time, perform the clinical study.
Yes, in the same context, as you remember, I think you forgot, we have isolated a protein of 60 kilodalton that is responsible to decrease glycemia. So we have the protein which is isolated and exerts anti-hyperglycemic effects, and commercialization is a different thing because it requires controlled studies in human beings, and perhaps in the next project. Yeah, you said about the natural food, natural food. Well, fat is fat. Fat is always bad in excess. The problem is this, it's a very economic problem. The more you eat, the better you become. We are in a world where we don't do a lot of exercise. We don't walk, we don't run, So this is a question of just the consumption of energies, yes. But what we observe, whether we are genetically obese, like children suffering from Karolinska syndrome, or in animal models like mice, whatever is the case, once we develop obesity, CV frequencies goes down, and that increases our palatability for fat. That is the problem. But if you are in excess of fat, whatever is the source, it's bad for you. It decreases CV frequencies at that time.
Thank you. Oh, you have an addition, okay.
No, I have a question for one of my colleagues.
I will allow it.
So I was wondering, the mutation in the receptors that you showed in this disease that I had was that in the homozygous context, or was it enough for the heterozygous mutation to cause the disease? I mean, if it's heterozygous, you wonder, is it dominant negative?
You mentioned the mutation of the captain B.
Exactly, that's the one.
Yeah, even heterozygotes cause it. Yes.
So the healthy allele doesn't have
Okay, another question. I have 3 hands, and then we can close this session. Oh, there's 4 hands. Oh, okay, that's 5 hands. So I have 5 hands before I close this session. I think I've noted the hands.
My name is Chidoke Abu and I'm from Nigeria in affiliation with Alex Epeme Federal University, Ebonyi State. So my question is to Professor Naim Khan, and he talked about the taste of fat and its implication in obesity. So my question is, do you think, or to what extent does psychosocial factors like stress and socioeconomic factors and cultural norms influence the taste perception of fat and its association with obesity in African populations study?
I'm so sorry, I could not follow your question.
Socioeconomic factors affecting
psychosocial factors. Do you think it's affected the influence of the taste perception of fat and its association with obesity in the African populations you study? Psychosocial factors like socioeconomic stress, status, stress, cultural norms, and all that.
Yeah, this is a very interesting question. I have 2 responses. What we are witnessing in Europe, that obesity is increasing, particularly in a segment that is living a very poor quality of life. I have seen in hospital consultations the women who have got no job, no love, nothing, no affection, they are getting obese. It's because of stress and corticosteroids, and they prefer to buy some food that is high-fat food, but low-quality food, and they take the enjoyment just by eating and by eating. That is a different story. But it has been seen in a study conducted in New York, in the outskirts of New York, and in a well-off family of the New York. And one colleague of mine from Monel, she did genetic polymorphism, and she said that the children who were determined to be obese because of genetic polymorphism in 2 populations, children from rich family were not obese. And then she asked what is the reason, that they were well informed that excess of fat and excess of sugar is bad. So the information and communication is very important in this sense, and family education. Even we did studies among the children 3 and 4 years old in the creche in France. We found that these children are given beans with fat, without fat. They always prefer with fat because they are supposed to eat the same thing at home. So family information and communication about food quality and well-being is very important, even though we are genetically predisposed.
Thank you.
Thank you very much. My name is Abayomiki from the University of Ibadan, that's in Nigeria. I have two questions, one for Professor Kubo, the other for Professor Anna. First, I'd like to commend Professor Kubo for a beautiful lecture on an otherwise abstract topic. Because anyone who has ever stood in a lecture where they're talking about ion channels, if you're not imaginative, you may not understand what they're saying. So I'd like to commend you on that. My observation and question goes first. In your presentation, most of the channels that you seem to have investigated in details are sodium and potassium ion channels. But what happens to channels like calcium, magnesium, ATPase channels? Are they not as important as sodium, potassium channels? Don't they exhibit the variety that you have studied for sodium and potassium channels? I just need to understand why haven't you studied calcium and magnesium channels as well? Can I ask the second question, or you want to answer that?
No, ask the question.
Okay, to Professor Anna, I noticed you studied pre- and post-exercise modifications in non-diabetic patients and in diabetic patients. And I observed that most of what was studied was one bout of 30 minutes exercise. First, I'd like to ask, why 30 minutes? Why one bout of 30 minutes? Why not one bout of 10 minutes? And why not one bout of 45 minutes like that? Secondly, given that the benefits gained from exercise are often derived from repetitive exercise, don't you think that if you were to study post-exercise modifications after repetitive exercise, you may see a better adaptation in type 2 diabetic patients? That is, their response to exercise may improve. Thank you.
Thank you. I hope the answers are more brief than the questions.
Let me first confirm your question. You want to ask the importance of other ion channels, which I did not introduce.
Why those ones aren't studied, and why this focus on sodium and potassium channels?
I see.
Sometimes the voice sounds
very simple answer is no strong reason. And I think all types of ion channels are as important, and all channels are worthy for study. But we cannot cover all types. And if we find something very interesting, for example, abnormal ion selectivity or non-canonical voltage-dependent gating, we want to dig it. But we cannot cover all types of ion channels. But please understand, all types of ion channels are as important for our function.
So I'd say thank you for great questions. You're very observant. So why pick 30 minutes? It's a brilliant question. 10 minutes is too short. 45 minutes is very hard for the patient. So it's basically one of these things. 30 minutes we know most people can do when you do 85% of heart rate. So that's why the 30 minutes. And we know that 30 minutes of exercise will give an effect on glucose control for the rest of that day. Then the question, why not a training study? Of course we should do a training study. And we should do different kinds of exercise. This was biking exercise. And it's just a matter of, we wanted to do all these rather, or very, very expensive omic analysis, so we wanted to really control. We wanted to know, what did the patients eat, when did they last eat, and take the biopsies exactly at the right times, at the right time of the day as well, so there's not there. And you just can't control people that well. It just becomes so difficult. So this was the easy way out, in order for us to get enough N numbers. We have tried to do some training studies, but people get ill, they forgot to go to their training, they have to go away for a Christmas meal. But those studies need to be done. They need to be done. Just because they're difficult doesn't mean we shouldn't do it. They need to be done. But we start with a low-hanging fruit. I think that's what we were told as a moment. Start with the easy ones.
My question is directed to Professor Kadire. You talked about filtering the fruit, the juice. What kind of filter do you use?
That's its secret.
I love that. More answers like those. It's my turn. Okay, thank you. I asked the professor about iron.
Closer to your mouth. Closer to your mouth. Closer.
My question is, how does the disturbance in ion channels will cause diseases? What's the mechanism? It's mainly because it's a distribution in the cell volume or cell excitability and action potential.
Sorry, could you repeat your question?
It's my question about that when we have mutations in the ion channels, that the disturbance, the mechanism of disease is due to change in the cell volume, because we know cell volume is highly regulated by ion movement, or because it affects the excitability and the resting potential of the cell.
Yeah, in the ion channel mutation associated diseases, so there are various reasons. So for example, if there is a mutation in the voltage sensor, so the voltage-dependent gating is hampered, but also by the mutation in the selectivity filter, there is abnormal selectivity. And also there are other types of mutations which affect trafficking to the membrane. So surface expression to the plasma membrane is hampered. So that's why, depending on the type of mutations, the reason why the disease is caused is varies. Did I satisfy your question?
Yeah.
Thank you. 30 seconds.
Okay, my question, I heard two concepts now. Professor Nan and then Professor An. Okay. So the concept I heard is one, olfactory into you. So olfactory. So when someone, a child up to 8 years, gets something or tests something, and then they try to develop test memory. Am I correct? And you say that the fetus or a child born to a pregnant diabetic mother. Is that you or someone? A pregnant diabetic mother becomes later on obese, and then you say that is metabolic memory.
Yes.
You see, they are overlapping concepts if you carefully watch them. Is it really metabolic memory or metabolic plasticity? Number one, if you know. In the case of neural system, a memory, what does memory mean? A memory means that when neurons make some change in response to some stimulus, they make some change, and then they store information. What we call is the stored information, not the change. So, in a sense, that is the concept of memory, and the neural change is plasticity. In your case, in the case of, I mean, it should be, I don't know, into that. The second one is test memory and metabolic memory. In the case of metabolic memory, I suppose that when a child is exposed repeatedly to high blood glucose level, they make some change within the cell, and that change eventually causes obesity. Obesity is a kind of happening. So the real essence should be metabolic plasticity, and I accept that when we work on this concept in the future, plasticity and the memory, the same thing, somebody says plasticity, another says the memory. For you, you get one chance for calcium. For calcium, he raised the question, have you ever tried the photofluorescence like thiocyanate or the dyes which attach to calcium in order to study the real virtual gating mechanism of what for sodium you cannot do it, for potassium you cannot do it, you may not do it. So to my knowledge there are no dyes that can fluoresce from these ions, but calcium is known for fluorescence. It is excellent to track when they move down and up through fluorescent microscope and then see the gating mechanism. Have you ever tried that?
Well, at least we have like the fourth congress, eh? This fourth congress topic. I have one, that was the last one that I have. I am told I have to move on, but further discussions can go on another time.
Thank you very much. I have two questions. One for Professor Anna and the second one for Professor Kubo. So Professor Anna, I'm interested in knowing the characteristics of the diabetic patients that we use. So were they, before they were put on medication for diabetes, were they maybe 20 years diabetic? And or was this effect that you found after exercise irrespective of their status? And Professor Kubo, about, I think generally protein specificity of interaction is very interesting. But in the case of ion channels, you talked about the size of potassium versus sodium being the most important way that these channels differentiate between the two. Is that the only mechanism? And bearing in mind that there are like the divalent method transporters, which can recognize several divalent ions. So what are the other mechanisms of specificity?
Yes, but just about the first question. Regarding the learning of taste qualities, it has been proposed by the scientists who work on the development, child development. They say that up to the age of 8 or 9, this is called a critical age. And there are many changes in the brain. Of course, it always happens. According to the theory of Hebb, every time there is a plastic new connections, some of them are regressed, some of them connected, but up to the age of 8 to 9, these connections are involved in the creation of gustatory memory. But it doesn't mean after 9 or 10 years we cannot change it. For example, I went to Beijing and I never ate grilled scorpions. Then I was watching the people eating grilled scorpions. And I was afraid, how can I eat them? But they were enjoying it. I said, let me try. I put it in my mouth and I said, it's not a scorpion, it's something different. And I enjoyed it. But I was looking in my memory, it resembles something I ate 10 years before in London. So I kept the memory of something, but I have to collect the gustatory memory, like in the hard disk of the computer, and I change it. Is it the same or is it the same? Now, I create another memory. Next time I will be invited to eat grilled scorpions, I know what is the taste of a scorpion. So I corrected it. But by the point of view, critical development of the child, gustatory qualities are encoded up to 8 to 9 years. That's why this is the age that's very important for the sake of obesity. And as Dr. Kadiri said, that macrozoic children, they are supposed to be up to the incidence of 50%, one after the two, to become obese or insulin resistant at the age of adulthood. So this memory is very important. That's why the gustatory memory up to 8 or 9 years is important, and obesity among children is very important parameter that we always see.
So my question was, what was the general characteristic of the type 2 diabetic people that we studied? And they were not completely newly diagnosed. Our exclusion criteria were, we did a cardio stress test on them to make sure that they could actually do the biking exercise. Nearly all of them were on metformin, but not on the day that we tested them.
metformin is something that nearly every single person with type 2 diabetes is on. We excluded people who are on any other type 2 diabetic medications, but we had to keep metformin in or we wouldn't have any patients. And then, because the person with diabetes sees their doctor more often, they actually had better lipid status than the control group, who didn't get all their lipids seen to. Blood pressure medication was about the same in both groups, but it's, you know, when you study people with type 2 diabetes, you start out thinking, I will have this perfect study and nobody will be on any medication, but then you won't have any patients. So we have to adapt to real life a little bit. But it's a very, very relevant question.
Okay.
I think two questions for me are somehow related, so that's why I'd like to answer in total. And the second question about the importance of divergent cation channels, is that correct? So yes, in addition to sodium or potassium channel, divergent channel, cation channel, especially calcium channel is very important. So the concentration gradient of extracellular, intracellular calcium is more than 20,000 times. So as you know, calcium is a very important biological factor in the cytoplasm. So that's why the study of calcium channel is as important, very important. And to answer to the first question, so the monitoring of calcium concentration in the cell is very important using calcium fluorophore or protein theta to ion channels. By using this kind of fluorophore, we can track the very local calcium concentration, for example, in organella or subplasma membrane, and or in the total cytoplasm. So these are very important approaches, and we also use it sometimes.
Amazing, amazing, amazing. Okay, so we're gonna modify our thunder clap, okay? Now we're gonna involve a little bit of raindrops before we start, okay? So we're gonna start, and there we go, and there we go, and thunder clap. Thank you very much, oh dear speakers, thank you. Time to stand before the WHO has a word to say to me about making you sit for all this time. So we're gonna stand for 10 minutes, have your tea, and be back here 1:45. Please don't forget to scan this. This has your book of abstracts. We'll be back. It'll be 2 rooms, and Asha Bilan will be able to explain what each room will be would encompass it. But let's be back 4:45.